and welcome to my channel Biology by Katie. In this video series I'm making my way through the A-level AQA Biology specification topic by topic. Today's video is based on Unit 1.6 adenosine triphosphate or ATP. As always, I hope you enjoy the videos, find them interesting, useful and easy to follow along with. Starting with the structure of ATP. On the screen you can see a single molecule of ATP, adenosine triphosphate. Now ATP is made up of three major components, or three different types of molecules. So we've got adenine first of all. Adenine is one of our nitrogen containing bases, our nitrogenous bases. So we've seen that before when looking at DNA and RNA. It's present in both. Adenine is present in ATP. We've also got a pentose sugar. Again, we're familiar with pentose sugars from looking at nucleic acids. We are familiar with both ribose and deoxyribose. Now, the pentose sugar present in ATP is ribose. Remember that pentose sugars means that they contain 5 carbon atoms. The final component of ATP, clue in the name triphosphate, is we've got 3 phosphate groups. And each of those phosphate groups is held together by a high energy bond. So that's our final ATP molecule, adenine, ribose, and 3 phosphate groups. So ATP is considered to be a really valuable energy currency molecule inside cells because of the fact that it can be easily hydrolyzed to release small, manageable quantities of energy that can be used to fuel many different processes inside cells. So ATP is hydrolyzed in a way that we're already familiar with from topic 1.1, the addition of water molecules to break apart bonds, in this case the terminal phosphate bond, releasing a single phosphate molecule and also releasing a small amount of energy in the process. So what we're left with is a molecule that has an adenine, a ribose, and only 2 phosphate molecules. So we refer to this one as adenosine diphosphate, di being the prefix for 2 rather than 3. And we also have the phosphate that was removed, and we refer to this as a single inorganic phosphate molecule, or just inorganic phosphate. This is a hydrolysis reaction and it is catalyzed by an enzyme called ATP hydrolase. Now, the overall reaction of this is ATP plus H2O, which is going to give us the products that are ADP, inorganic phosphate, and during this process we also have energy released. So now that we've seen how ATP is broken down, it's important to recognise that this process is a reversible reaction. So ATP can be resynthesized using the monomer unit, so using ADP and an inorganic phosphate. And if you remember from the very first topic about monomers and polymers, we know that when we build a polymer from monomer units, this is a condensation reaction. So it involves the removal of a water molecule, a single H2O. This condensation reaction resulting in the formation of ATP is catalyzed by a different enzyme called ATPase. And this process usually occurs during photosynthesis, during the light-dependent stage of the photosynthesis reaction, and also during respiration. When ATP is hydrolyzed into ADP and an inorganic phosphate, the important use of this process is the fact that the small manageable quantities of energy released are then used for various different mechanisms. However, we do also have a use for the inorganic phosphates that are released. Adding phosphate groups to other molecules such as enzymes and proteins actually causes them to become more active or reactive. We call this process phosphorylation, and it often leads to the activation of enzymes and proteins. For example, this process of phosphorylation happens during the first stage of respiration, glycolysis, where 2 phosphate groups are added to a single glucose molecule. This in turn makes it much more reactive and kickstarts the series of reactions that follow. So just to recap, the major value of ATP inside living cells is that they release small manageable quantities of energy when they are hydrolyzed. And the inorganic phosphates from which they are made can also be used to activate enzymes or proteins within cells. So just having a look now at the points on the AQA specification for Unit 1.6 ATP. The first one is a single molecule of adenosine triphosphate is a nucleotide derivative and is formed from a molecule of ribose, adenine and 3-phosphate group. So we covered that when we looked at the initial structure of ATP. We also have the point that when hydrolyzed, ATP is converted into adenosine diphosphate and an inorganic phosphate group, and this particular reaction is catalyzed by ATP hydrolase. The inorganic phosphate released during the hydrolysis of ATP can be used to phosphorylate other compounds, often making them more reactive. So that was where we discussed the idea of phosphorylation of things like enzymes, other proteins, and of course glucose was the example we used. And the final point on the specification is that ATP is resynthesized by the condensation of ADP and an inorganic phosphate, and that this reaction is catalyzed by the enzyme ATP synthase. And of course, this happens during photosynthesis and respiration. So we've covered all 4 of the points from the specification in detail, and that is topic 1.6 ATP finished.